So good afternoon, everyone. Welcome to this new episode of the SDDN webinar series, the Spanish Drug Discovery series. That today we have the pleasure of co-hosting this with Eric. So let me introduce the association for you. So SDDN is the Spanish Drug Discovery Network. It's an association of professionals working in discovery and development of new therapies. Although it was born focusing on the Spanish community, the vocation is moving and operating internationally and inviting people abroad also to join the association. So we have individual members and we have entities, companies, which are partners of the association. So it's a melting point. SDDN is a melting point of professionals from academia to industry, business, research, technology providers, and pharmas. So we have a mixture of all the professionals. We have a voice in this field of drug discovery and development. So we are very grateful to our partners. You can see here our partners last year. So if your organization wants to join us, we're more than pleased to have it on board, to have you on board. You can go to the web site. Actually, I mean, you can scan this code or go directly to the website to join the company, the association, either as a member, individual member, or as a partner. You have benefits that are described in the website. So this is a high-level summary of the SDDN activities, what we are organizing for our members and partners. The annual meeting is the flagship. I say a word later on. The flagship of the society of the SDDN. We have the SDDN webinar series as the one we are attending today. SDDN cares a lot about young professionals, right? Early career professionals, and we are trying to support them in their career. And one of the initiatives is this mentoring program. We have just started the second edition in January. So we have in the website, you have access to the capacity map, who is who in Spain working in drug discovery. And we have the SDDN newsletter. So if you join the society, so you can have access to all this information. So the annual meeting last year was in 2025, was held in A Coruña. We brought records in terms of figures, in terms of number. Just to give you a flavor of the size, we have 170 people registered with 25 sponsors exhibiting there next to the auditorium, next to the speeches, and we have 35 posters, 3 scientific tracks. So we were successful last year. Hopefully we will renew this success this year in Pamplona. So in October, late October, is where the annual meeting will take place. So we invite you to join. So we are about to launch or to announce the save the date. So please follow us on the website and LinkedIn, and to keep you updated on when the registration is open. And now I'm going to hand over to Sanjay Kumar, the CEO of Eric, who are kindly supporting SDDN for this webinar, and also as event sponsors in the past. So Sanjay.
Thank you, Julio. It's a delight to be here. Thank you so much. My name is Sanj Kumar. I'm the CEO of Elrig, which is a UK-centric organization, but it has very, very, very similar aims to SDDN, and that is why we're working together. Plus, Julio and I have known each other for more years than we care to count. I hope we're not a stranger to all of you out there. But if we are, if you've not heard of LRIG, this is basically who we are. We are a learned association, and we serve the drug discovery community very similar to SDDN. As I said, we're UK centric, but we do spread our wings a little wider than the UK. We have meetings throughout Europe, and actually this year we're also going to the US, to Boston as well. We're not for profit, so that means we put all our meetings on free to attend. You can see these are highlighted in the words here. So if you would like to come to an LRIG meeting, you can come, and all you'd have to do is to pay for your travel and your accommodation. There is, even though you have to register, there's no ticket price, and indeed we'll even provide refreshments while you're there as well. Our community, we've been going about 25 years, and so our community is about 22,000 what we call early drug discovery researchers. So these are people in target validation, assay development, high throughput screening, and then lead optimization. That's the group that we serve, and that's how we know Lyra, who we'll introduce in a short while, because we work very closely with the pharma, biotech, and CRO communities. How can we be free to attend? Well, all our meetings are supported by life science suppliers. We run associated trade shows alongside each event, but that's great, not just because they provide sponsorship, but also because they provide the tools and the technologies and the applications that we all need in our drug discovery world to get our research done. So that's who LRIG is. And on the next slide, Julio. Thank you. These are the events that we're doing in 2026. I won't go through this like SDDN. You can go to our website, elrig.org. It's down the bottom there on the left-hand side. And you can see that we put a number of events on throughout the year, and they're in different countries as well as different topics as well. If there's one event that I urge you to come to, it's the one in the red tab. It's in October. It's I think 2 weeks or a week and a half before yours, Julio.
Yes.
It's in London. It's called Drug Discovery. It's a 2-day meeting in Excel in London, and we all know that Spain to London is relatively easy to get to. You can certainly pick up a Vueling or an EasyJet or even a Ryanair flight, relatively inexpensive. You can even take the train as well. And it's in the Excel Centre in London. At that meeting, there will be 3,000 delegates. I'll say that again. There will be 3,000 delegates, all from the early drug discovery industry. There'll be over 250 exhibitors, and this is the important bit, over 150 talks. Will also scientific talks will be taking place across 16 tracks across 2 days, and they're on all sorts of different topics. And that information you can see on our website. And I'm sure Lyra and Julio will both be there, and you can meet them face to face as well. And I just want to end, and this is just my last slide, is really to say that we're looking forward to 2027 now. I'm really delighted with the relationship that LRIG and SDDN have. And Julio and Jorge and I are talking about now running a joint meeting together to take place in 2027 in Barcelona in June. So LRIG will be coming to Spain for the first time and working alongside SDDN, and I'm really looking forward to that meeting. But more details on that as we flesh that out. So Julio, thank you for this. And
yeah, that's terrific. You are more than welcome. We'll do our best to host you as you deserve. So
Yeah.
next year. So we are looking forward to working together on that event. Yeah. So today's lead role is Larry. So Larry, the virtual floor is yours now. You can introduce yourself, and we are looking forward to listening to you. I'm going to stop sharing, and then let's see if I can. Yeah. Yeah. I think it's up to you now.
I'll start sharing now. Let me know when you see the slides coming. Can you see that online?
Yes.
Yes,
Perfect.
no. Yeah.
Perfect. Okay, so yeah, thank you, Julio, for connecting me to SDDN, and Sanj for supporting from the ELRIC perspective. It's great. It's great that you've invited me to this webinar. I have a long relationship with ELRIC. I've attended all the ELRIC events since I've been in industry, and Julio and I met each other at GSK, so it's great to reconnect. And yeah, thank you all for joining. I hope this session is informative, and we can take questions at the end, and I hope it's relevant to the work that you're doing. So my name is Leire Escudero Ibarz. As Julio mentioned, I am an associate director in the Discovery Sciences department in AstraZeneca. And within AstraZeneca, I work within the functional genomics team in the Center of Genomics Research. And today, on behalf of the team, I will be giving a presentation around our functional genomics capability to identify and validate novel targets in AZ, and particularly focusing on the challenges and opportunities that we find in the biopharma space. So this is the presentation at a glance, quickly, so that you know what to expect. Julio asked me to describe my scientific journey in case it's helpful for that early career professional community that Julio mentioned, that SDDN is really keen on helping. So yeah, I just have a slide on myself. The rest will be science, I promise. I will then move into emphasizing the importance of target identification and validation in the drug discovery process, and describe some of the platforms that we have built in AstraZeneca to enable this. I will also focus on our functional genomics arrayed CRISPR screening capabilities, including specific examples around the immunology and respiratory space, which is what we call biopharma. And hopefully at the end, we can sort of wrap up with some future opportunities that might be of interest in this community and in this field. So in terms of myself, as I said, this is a very egocentric slide, but hopefully it gives you a bit of a flavor of my journey. I am originally from Spain, from Barcelona specifically, where I did my BSc or licenciatura in the University of Barcelona, in the Autónoma de Barcelona. I also spent some time during my degree in Italy, in Università di Pavia, where I did a placement, and this was, well, really wonderful experience to be in Italy. I have a special attachment to Italy since then. And yeah, following my degree, I just decided to go abroad. I was not so sure where to go, but in the end, after talking to mentors and people from the department, I decided to continue my journey in London. So I moved to London to pursue a master's in molecular pathology and virology in Imperial College London, and spent some time in the sort of gene therapy area using viral vectors in motor neurons. So essentially at that time, my plan was to return to Spain. I was just going abroad for a year, but as you know, life takes you in different directions and turns, and I've not left the UK. So I've been in the UK for 16 years now and counting. I have no plans to return, not yet. But yeah, I mean, the UK has really given me a lot of opportunities to expand my role. So yeah, after my master, I was actually not 100% sure if pursuing a academic life. So rather than jumping straight into PhD, which is what we normally do in academia, I spent some time in King's College London, working in a lab to just get more experience and hands-on lab. And I was actually in a laboratory that was focusing on aging pathways using nematodes and worms, which was really different to what I do now. And at that point, I decided that although I wanted to eventually move to industry, it was really important for me to pursue a PhD to sort of complete the academic puzzle. And I moved to Cambridge at that point. So I started a PhD in cancer genetics at the pathology department, which was quite clinical, one translatable. And I felt that I was getting closer to the patient at that point. And the beauty of Cambridge as well, and during my time doing the PhD, is that Cambridge is an ecosystem that has a lot of opportunities outside of your core role or your day-to-day role. So I did quite a lot of activities outside of my PhD. At that point, I engaged with the business school and the entrepreneurial community in healthcare, which is really innovative in this area. And that led me to co-found a healthcare startup, HEALARC, which essentially I sort of juggle in parallel to doing my PhD write-up. And then eventually I decided it was either going full on the startup or jumping to industry. And at that point, I decided to do the move to industry. I joined Horizon Discovery because at that point, the CRISPR as a technology for target discovery was essentially booming, and Horizon was pioneering this. So I decided to go to Horizon, learned loads of CRISPR and cell engineering in that department, and that actually led to the future roles that I've had in industry. So after Horizon, I moved to GSK, a pharma company close to London. Where I first met Julio, in fact, and that was my first sort of job in big pharma. And essentially, I was using, I was part of a team that was using CRISPR and iPS derived models and technologies to be able to support multiple projects in the drug discovery pipeline. So as you can see, I was never associated with a specific therapy area, but I've always been in departments that uses technology and cutting edge science to advance different projects in the drug discovery area in different therapy areas. And yeah, after GSK, I then moved to AZ, where I'm now. As I said, I'm in the functional genomics department. I will give you, during the presentation, you'll see what we do at AstraZeneca in this space. And I feel really fortunate to be part of a group of scientists, as you can see here in the picture. We don't all fit in that picture, but some of them are there that come from, you know, a diversity of sectors and backgrounds. And although we all bring different things to the puzzle, we all focus on discovering novel targets for different therapy areas and also for cell therapy strategies within our company. At the bottom, I actually wanted to highlight for those that are not in pharma and might be interested in being part of pharma. I just wanted to sort of emphasize that pharma actually allows you to wear different hats within your current role. So although at the moment I'm responsible for a team that drive those CRISPR screening and other novel target efforts in the company, we wear also different hats. So I'm also part of I work very closely with the immunology group, so I'm responsible for the immunology portfolio and strategy on behalf of the team, which gives me that sort of matrix working approach across different teams. Also, AZ encourages you to take secondments and learn from different teams, which is really exciting. So, for example, last year I was in a secondment in the Global Project Management Group, which gives you exposure to different parts of the drug discovery pipeline and governance. And also, AZ allows you to be connected to the external world, the external community, and collaborations with our partners. And this is a good example, right? Being connected to organizations like SDDN and ELRIC. These are things that are very much encouraged. We also have a lot of collaboration with postdocs and students that come to our labs, and we are part of mentoring networks within the sort of Cambridge ecosystem. And also I wanted to highlight and advertise a little bit that I'm part of a non-profit global organization called HBA, which ensures the acceleration of leaders in pharma. So if you're interested, actually there's a branch in Spain. Barcelona for sure has a branch. I'm not sure if Madrid has one as well. But if you're interested in HBA, it's a very good community to join in. Right, so now enough of me. Let's get into the science. And I wanted to start by highlighting why target discovery is so important for us. And I think this is obvious to all of us and to the community, because one of the biggest challenges that we faced as a field in healthcare and in pharma is to be able to identify the right target that is linked to the relevant disease. So the stats say that 90% of drugs that enter clinical trials actually fail, and most of drug projects fail due to lack of efficacy. So essentially, meaning that the target we have chosen at the start of our drug discovery pipeline is not really driving the disease in patients. So in order to improve that R&D productivity, AZ has been focusing on on making our decisions based on 5 technical determinants that are part of this 5R framework that was published some years ago, where we essentially focus on the right target, the right patient, the right tissue, safety and commercial potential to drive any drug discovery project. And this is and has been really much embedded in how we work as a team and how we drive the right culture internally for that scientific rigor and ensure that we are successful with our drug discovery pipeline downstream. So overall, if you look at this, this continued evolution and the application of this 5R framework are really beginning to have an impact, improving the success rates in the clinic. But really, selecting the right targets is still the single most important decision that we make early on in the drug discovery process. Is the first step, essentially. And this is because the primary reason, as I said, for this failure is that that the candidate drug in clinical trials is not it's not really down to only the safety or the chemistry, but actually the efficacy of that drug. So there is the need to reduce that clinical attrition by driving this sort of demand of discovery of novel genetically validated drug targets in the right translatable in vitro models that we model in the dish. Right, and to continue on this theme, I wanted to sort of share with all of you how AZs have invested in this, how we've invested in novel target discovery. And we have invested in multiple platforms for target discovery in order to deliver those novel and validated targets in multiple disease areas and drive those portfolio decisions that get us to select robust candidates. And for this, I really wanted to emphasize that all these parallel platforms that you see in this slide, which includes, as you can see, human genetics, omics data, in vitro and in vivo models, and most recently AI, which, as you can imagine, is completely revolutionizing how we are driving our pipeline. These platforms are actually more powerful when we integrate them all together, and therefore the collaboration across all of these teams and disciplines is really crucial, and we have a matrix team focusing on shared goals to get there. So the design of this pipeline you see here, from data generation to target nomination, really depends on that complexity and feasibility of our in vitro models for screening. And really the biology that we are trying to interrogate, which is, as I mentioned before, fully linked to the disease area that we're trying to tackle, and also relies a lot on the available models that we have in this space. So thinking about this, it's for me, target discovery is an engine that has two halves. So first we have to identify those targets and then validate them, starting from that data generation with the large data sets that we have from genetics, omics, etc. And then we augment that information using AI, and that leads us to the nomination of target candidates that we test in vitro and in vivo to select the best candidates that will make it through the pipeline. Oops, sorry, I skipped one. So this is the vision of functional genomics, which is part of that big engine that I was describing. Our vision is that we apply our expertise to enable that world-class portfolio in discovery projects through the identification of novel targets and clinical insights and mechanisms, and that biology that allows us to enhance our understanding of disease biology, and therefore hopefully increase the probability of success of our drug candidates in the clinic. So the functional genomic department focuses on that novel target discovery by interrogating those targets in physiologically relevant models, and we use a suite of genetic and non-genetic perturbation libraries, leveraging the power of human genetics and data science as well. And this is a bridge to sort of introduce you to what functional genomics is and how we are distributed in the UK in AstraZeneca. We have 3 key CRISPR screening platforms and teams, or locations, if you wish. We have on the left-hand side our core functional genomics screening group within AstraZeneca, which focuses at delivering genome-wide and bespoke arrayed CRISPR screens for all therapy areas. So we cover oncology, biopharma, which is everything that is non-oncology, and most recently rare disease, since Alexion joined AstraZeneca. Then at the middle, in the middle box, we have the Functional Genomics Center, which is a collaboration with CR UK and our oncology therapy area, which focuses on delivering genome-wide pool CRISPR screens rather than arrayed. And this is to identify mechanisms of sensitization and resistance to different oncology indications. And the last but not least team on the right-hand side is actually formed recently. So this is a newly formed functional genomics screening laboratory, which is a joint collaboration with AZ, the Medical Research Council in the UK, and the Milner Institute, which has been operational from early 2025 and essentially gives us access to translatable disease models for arrayed CRISPR screening as a collaboration with academic groups in the biopharma space. So we don't run oncology projects in this center, but it's everything that is non-oncology in collaboration with academics that submit proposals. And I have a slide at the end of the presentation where you can look at the call for project proposals if you are interested to submit a project. Right, so as this is quite obvious to all of you, but the functional genomics platform essentially investigates the link between the information contained in our genome, so in our genes, to the functional effects of this information in disease. So for me, it's like connecting that black box between genes and the genotype and their effects and their phenotype in healthy and disease backgrounds. And I guess the workflow for us starts with defining what is the biological question and generate those hypotheses. And that happens in collaboration with our therapy area experts. We do that by identifying targets that can be interrogated using our phenotypic arrayed CRISPR platform, which is the one that I described before on the left, which is the sort of core unit in AstraZeneca. So this is how our toolbox looks like, and this is a very surface representation of our team, but essentially, in order to maximize at-scale screens in functional genomics for phenotypic screening, we combine the use of translatable disease models, as I said before, and that includes from primary cells to more complex 3D and co-culture models that we can then couple with predictive high-throughput assay readouts, such as high-content imaging, flow, cytokine release assays, anything that you can read out in a 3 or 4 well plate, essentially, we can read. And we then combine those with cutting-edge perturbation agents that, as I said before, genetic and not genetic, that can interrogate those models, so perturb those models. You will see that we are mainly focused on CRISPR editing, but we also use other libraries like secreted protein libraries, compound libraries, and so on to interrogate those systems. And the scale that we use forces us or encourages us, let's say, to integrate all our capability with automation. So we have automation experts and automation scientists in our team that allow us to do this at scale, because as you can imagine, it's something that we could not do manually. And also we collaborate closely with our data scientists, because we create a lot of data out of this screen. So it's very important that we not only look at the data downstream and try to interpret and analyze and interconnect and integrate in our AI engines all of that data set, but also we do use data teams to enrich and interrogate relevant biology at the beginning of our screen, so that we can actually funnel the right targets at the beginning of our screening cascade. And once we've identified those targets, we move into target validation, which of course increases the mechanistic insights on certain hits from our screening cascade. We can look at opportunities for biomarker evaluation that essentially allows us to accelerate those projects through the drug discovery pipeline. And then we collaborate with the post-target identification teams to ensure that those targets make it through the drug discovery pipeline. And in terms of impact, although you could think that the impact we bring to the field and to the company is to identify new drug target hypothesis, which is true, we focus on that novel target discovery, we are also adding impact into other areas like combination strategies, for example, also different strategies to armor our cell therapy products, thinking about different modalities, and increase that mechanistic understanding of some of the drugs that are already sort of being developed by our partners. And here I wanted to highlight briefly the key challenges that we find when we build these at scale array CRISPR screening workflows, and how that increases the complexity when we use disease-relevant in vitro models. So we have actually shifted most of our projects into using primary models that are difficult to use, because that's what our partners want. They want models that mimic and reproduce the disease in the dish, essentially. So these disease-relevant models present constraints of cell availability. So sometimes, these are models that come from a patient isolation, and we struggle to actually get enough cells to actually run a full CRISPR screen, because they have limitations of growth without affecting the phenotype. Also, most of these models are refractory to delivery methods of CRISPR that are amenable to the scale. So we have worked a lot on different delivery methods to be able to get those CRISPR reagents into those cells that are that complex. And as you can see in this image, essentially, we see how scale and translation sometimes are on the extremes and in the polar opposites. It is true that now we're getting better at scaling translatable models, but we always need to think that it's not the same scaling a cell line versus an organoid, where cell availability and delivery of reagents might be a problem. But essentially, we have been working on this. We have been building different capability to continue innovating in this space. And the other challenge, well, maybe this is not a challenge, maybe is a comment on our scale, is that the scale, initially, we would see it from the number of perturbations that we were introducing into those cells. So if you think about, okay, if we do a whole genome screen, we will have 20,000 genes, which means that we are using 20,000 wells at scale in 384 well plate format. So the scale can come from that. But actually, we are seeing now that we are also increasing that scale, not only from the perturbations, but also from the complexity of our assay conditions and endpoints. So we are mimicking the disease in a way that we have different stimulations in our cell types and different endpoints that we want to collect to have those unbiased results that allows us to interpret the data. And in this context, the way we see it is like these pyramids that you see here. So we can look at the scale and high scale by thousands of genes that normally come from either whole genome of knowledge graph predictions that we funnel into one specific biological mechanism, meaning one cell type with one assay readout, for example, imaging. So that then you focus your scale in the perturbation, but you actually funnel everything in one specific assay, which gives you that assay depth. But most recently, we've been working on a lot of those human genetic targets that have a very clear human context. So these are human genetic targets that are enriched in disease in our sort of disease cohorts. And therefore, we do know that are really important targets that come from whole genome sequencing and rare variant analysis. We know they are important. We know that they hit the patient population. But actually, we don't know in which assays to funnel those genes, because we have no idea how they are acting in the body. So in that context, what we see is a shift to maybe narrowing down to less genes being screened, but our scale increases massively because we need to funnel all of those genes in different biological mechanisms, and therefore gives us that assay breadth and sort of complexity of the disease in the dish, so that we can start understanding where those human genetic targets are acting in the mechanisms in the disease and the biology. So yeah, that's sort of something that we are sort of evolving into as well. And the other aspect I wanted to highlight here is that sometimes those human genetic targets might be expressed in different cell models, that's important as well when we choose our model of interest. So we intentionally characterize the disease-relevant treatments and in our endpoints with omics in our experimental workflow. So essentially, we have a suite of cell models, which I will show in the next slide. We drive different differentiation into these cells with different treatments, etc. And then we use transcriptomics and proteomics to understand how those genetic targets are expressed into those different systems, because disease versus healthy might have a complete different expression pattern. And that allows us then to bucket those relevant targets by expression into the different screening models and cellular mechanisms, so that we increase the success rates of those targets going into the right assay. Otherwise, we won't get a hit from that assay, but it's inconclusive. It's not a negative result. It's basically we're not looking in the right model. And that's really important to keep in mind for us. And on that note, as I was saying, our arrayed CRISPR platform for biopharma specifically is evolving at high speed because we have really changed the ways of working due to the rapid rise of biopharma clinical research, which now has really fueled the demand for complex in vitro models that are able to mimic that heterogeneity of immune and respiratory disease. The complexity is increasing more and more and more every year. So looking at the diversity of those indications and disease cohorts that we have available on the left-hand side, coming from genetics or other information, omics sources, for example, we have now identified key in vitro models that actually capture the complexity in the dish and that have shared mechanisms that we can mimic in the dish. And that's how we have built our arrayed screening platform. So again, and this is a bit repetitive of what I said before, but this sets the scene for the next slides that I'm going to show some project examples where we have different perturbation libraries, so we can perturb these models that I mentioned with different libraries that include CRISPR, compounds, secretome, clinical antibodies, cytokine combinations, etc. And we can then use these ones to perturb the models itself, which, as I said, normally we see it in the primary cell and complex cell model front. And you can see here some examples that I'll show you in a minute where we are able to CRISPR edit at scale monocyte-derived models, T cells and Tregs, so regulatory T cells models, pathogenic fibroblasts, epithelia models, and most recently we're working on organoid models that allows us to sort of be closer to the in vivo situation. And as you can see on the right-hand side, we are able to integrate all of these models with different readouts, different multiparametric readouts that include high content imaging, which is fully automated and gives us beautiful images where we can extract a lot of features. We are also able to look at high throughput multiflow, where we use flow analysis for looking at different markers of expression in the cellular models. We have high throughput capability for cytokine release assays, quite pricey, but we're trying to improve that. And also we are able to start looking at co-culture models where you have the edited cell in one side and then you co-culture it with a relevant model that might be found in that specific disease ecosystem. And there's actually a lot of interest, as you know, in the field around omics. So we are trying to make sure that we can embed our omics readouts in our 384 high throughput capability as well. And that will allow us to have more of an unbiased readout that rather than funneling it into specific markers of disease. So in a nutshell, this diversity is a toolbox that allows us to capture this complexity of multiple disease indications. And this is what I'm going to show in the next slide. So I'm aware of time. So I have few case studies in immunology and respiratory. What I'll do is give you a flavor of maybe 2 or 3, and then maybe I'll skip some slides because I'm aware of running out of time. So let's start with the first one. There's quite a lot of information to digest, but I thought that it would be better if I give you a quick run through of things that we have been working on so that you get more granularity on what I've been meaning in the last few minutes. So these, again, I'm showing all these projects on behalf of the team, and all the faces that you see in the slides are the people really delivering this from the lab. We have been recently working on a project that aims to deploy our arrayed CRISPR screening platform to identify targets that are relevant to Treg stability and functionality, so regulatory T cells. This cell type is really important in immunology. Immunology is a therapy area of high importance and priority in our portfolio at the moment. And to be honest, it's not only immunology per se as a therapy area, but as you can imagine, immunology is present in all diseases at the moment. So we are interested in immunology as a whole. And I guess, why is this important? Why is it important to work on this project? So as you might know, dysfunction of regulatory T cells can lead to various immune-mediated diseases, and Treg stability declines under the inflammatory conditions that are found in patient tissue. So in order to overcome this challenge in autoimmunity, we need to find ways that allows us to boost the functionality and the stability of these cells. And we have been looking at leveraging our CRISPR editing capability in Treg for novel genetic target discovery and validation of targets that come from human genetics. But we have also been using it to create new opportunities for the Treg cell therapy portfolio. So using Tregs per se as a living drug that we can use in autologous or allogeneic cell therapy to achieve long-term remission in inflammatory disease. And how do we do this? As I mentioned, all of these workflows are based on our RAID CRISPR screening platform at scale. We use primary human naive Tregs, and we are able to cryopreserve them, scale them, introduce CRISPR reagents at scale, and then treat those Tregs that have been edited with different cytokine cocktails that, as I said, mimic that inflammatory environment in the tissue, and allows us to look at multiparametric readouts, including flow, cytokine release, Treg and T cell immunosuppression assays and proliferation and viability. So what is the impact of this? Of course, the clear impact here is that we are able to now, with the screening, identify targets that are hits of Treg stability and functionality. So those are the targets that the therapy area is interested in drugging with different modalities. And also, we can look at the intersection of the different endpoints, including immunosuppression with stabilization markers and inflammatory cytokine release, to look at the targets that are most promising to take forward. And also something I wanted to highlight in this slide in particular with the graphs that you see in the middle is that in functional genomics, we are now using the concept of high dimensional experimentation, which is able to look at multifactorial and combinatorial conditions in the dish, so that, using stats and mathematical models, we can predict the response in the dish of certain conditions without the need of having to test all of those parameters and all those conditions. So we appreciate that different parameters can actually have an effect in that heterogeneity of the disease. So it's very important that we can combine that at scale. And there was a very interesting project on Tregs where we wanted to mimic the disease-relevant ecosystem of a Treg in vitro. And it was really hard to actually identify which cytokines and which inflammatory conditions were driving that phenotype so that we could actually find targets around this. And the team was able to use high dimensional experimentation to enable this. So essentially, just to give you an example of scale, an experiment to combine all of the cytokines to get us there would have required 63A4 plates, so you can imagine the number of conditions there, manually, which was impossible for a human to pipette in. But we were able, with HD, we were able to reduce that to one single 3A4 well plate. So as you can imagine, for sustainability, we're reducing reagents, plastic, hours, time, efficiencies, etc. And we get a very robust readout where we are able to identify a treatment condition that mimics that disease environment. So that's been very interesting, and we're using HD now in every single project if we can, because it's been very successful. I'm going to skip this one. Here, I just wanted to say that the same as we're using regulatory T cells for the immune portfolio, we are also using, as you can imagine, macrophages and monocyte-derived macrophages in order to identify targets of macrophage polarization, because macrophage polarization is one of the key axes in disease pathogenesis that sort of modulate that inflammatory versus anti-inflammatory response into different patients with immune-mediated diseases. And I'm not going to dwell into this, but essentially, we can also collect different endpoints from CRISPR screens. We can look at knockouts that have an effect in different polarization stages of different donors that we look at in vitro, and we are able to identify hits that are relevant. And in fact, what is very beautiful is that, as you can imagine, for certain disease indications, such as IBD, for example, we can actually look at the hits from macrophages and Tregs and T cells and different cells that are actually actively causing that disease to see what are the targets that are more important. And I think I'm going to give two more examples. Julio, how am I doing it with time? If I give one or two and then we go into questions.
For sure, for sure.
Okay.
For sure. Yeah. No,
Otherwise, you have to
problem.
stop me. I can speak forever. Okay,
Yeah,
so
no.
I'll give you this example because I've given two examples in immunology. I wanted to also show you what we've been doing in the respiratory space. So we've been able to use long models of idiopathic pulmonary fibrosis, which is a chronic disease that causes lung scarring and fibrosis in the lung, which eventually leads to respiratory failure and is a very detrimental disease disease for patients. And the same way that we are able to sort of isolate primary cells for immune models, we are also able to source primary lung fibroblasts that come from patient donors, edit them at scale, and mimic a fibrosis as senescence phenotype using different stimuli. So for fibrosis, we use TGF-beta, and we look at levels of alpha-SMA expression, which is a marker of fibrosis. And we also use etoposide to drive senescence, looking at P21 as a marker of senescence. And what you can see here on the right-hand side is that we are able to modulate this system very well. You can see how TGF-beta induced expression of alpha-SMA is lighted up in green, and that indicates that we are driving fibrosis in these models, in these lung models by imaging. And you can see the hit knockouts at the bottom of the image are able to completely prevent that fibrosis. So what we're looking is targets that allows us to prevent the fibrotic phenotype in the lung. And this project is actually linked to this other project, and then I'll stop giving you examples where we have translated the lung fibrosis pipeline into intestinal fibroblasts as well. So we've seen that the fibrotic mechanism and the models that we build for fibrosis can actually translate across different organs. So rather than looking at lung fibrosis here, we're looking at intestinal fibrosis, and how intestinal fibrosis is one of the key mechanisms for IBD, or inflammatory bowel disease. And again, I'm not going to go into this slide, but we are able to identify markers that are overexpressed and have a detrimental effect in disease with different pro-inflammatory and pro-fibrotic conditions. So just 2 or 3 slides to wrap up. With these examples, I hope I have been able to show you how drug discovery is really a team effort. So we have to integrate multiple teams in both the in silico and in vitro discipline, different disease areas that includes external partnership with other industry and academic collaborators across the globe. So we are a global company, and it's not really possible to achieve those goals without that matrix team. These are the final insights, but I hope that with this presentation, you've seen how important that genetic linkage to disease and the translatability of the right models to get to the right target is, and how AZET is really investing in that end-to-end workflow to be able to fuel new targets into our portfolio. We've been doing that through our RAID CRISPR platform. We combine human genetics with the AI and knowledge graph technology to be able to identify novel targets and cell therapy strategies through our collaborative effort across different teams. And with this slide, I wanted to sort of highlight that, as I mentioned before, AI is really transforming how we do research at pace. I was in a panel discussion last week where I heard this analogy of AI being the new industrial revolution, and I completely agree. AI is really revolutionizing everything we're doing, and we need to constantly adapt. And this slide really shows how it's really important to integrate a set of capabilities around genetics, omics, and through partnership to be able to access that high quality human cohorts and patient data that come from diverse data sets. And this is really important so that we can actually get to those target insights successfully. This is a QR code for you to take a picture if you want around the Functional Genomics Screening Laboratory, which is taking now project proposals from academic collaborators if you're interested. I'm happy to share the details with Julio if he can share it with the community later on. But this is a partnership that we have that have been very successful so far. And yeah, last but not least, I wanted to thank everybody that has been involved in this work. This is not an exhaustive list. There's a lot of people involved that might not be here, but it's really a pleasure to work with everybody. And I thank you all for listening, and I'll take any questions.
Thanks so much, Larry. So really amazing.